Thank you so much for giving me the opportunity to be here. Thank you, Melanie and team, for organizing a fantastic conference. It's really nice to be an attendee this time and not an organizer like usual. So it's my pleasure to talk today about a project that we had in the lab. So I run the Center for Emerging and Neglected Diseases at UC Berkeley, and we were involved in the COVID pandemic with a variety of projects, really trying to help things along as much as we could. We've worked on such diverse things like hosting a hackathon to find, to assemble new ideas what to do in the early pandemic. We helped with a ventilator project. We helped to establish a testing capacity for COVID in our collaborator sites. Makerere University in Kampala, Uganda provided PPE. That was an adventure on its own. We also raised $1 million to give badly needed seed funding. Many of you have tried to do really groundbreaking research early in the pandemic, while NIH and other funders were horribly lagging behind in actually giving money out to do that important work. We came in, similar idea as FAST grants also had on a larger scale, give money without many strings attached early on in the pandemic, and that was really, really productive. Out of these projects that we funded, I did the math recently, writing the final report. We outperformed NIH by 15-fold in terms of output from these projects, having a very, very simple review process in the beginning, so I'm quite proud of that. And then we also helped develop a new method to do lamp-based assays, like many of those were developed in the pandemic, but we have a team that can now distinguish for $5 if you have flu or COVID, which is also quite nice, and they're trying to commercialize that. And lastly, we've also contributed to therapeutic discovery, which is really my area of specialization, because I have more than 10 years experience in the private sector doing preclinical development and drug discovery. And we published several papers on this, and one story I would like to talk about today. Idea we had is, instead of just screening straight up in the biosafety level 3 SARS-CoV-2 against all kinds of compounds, what if we could use what's already there, which at the time was remdesivir, which worked really well in vitro, and the clinical data was a little bit mixed, so we thought maybe we can improve remdesivir. And the strategy I devised here was a synthetic lethality method, which was to screen, instead of just screening for actives and combining them like in traditional combinatorial techniques, we would look at screening in presence of some remdesivir to generate synthetic lethality pressure, and to see if you can find compounds that are active only in combination. So that's really a method that is adapted from the yeast field, where this is commonplace and people are very familiar with it. Parallel pathways allow the pathogen to overcome a compound that interferes, and like that, if you knock out both pathways or you have something, then you can get a lethal effect. We tried to do that by in a collaboration that was really productive with Professor Sarah Stanley at UC Berkeley, who initially worked on tuberculosis and was really an expert in biosafety level 3 work, and Eric here and Scott Beering as well, who's in the audience today. And the idea was to enhance the efficacy of remdesivir through an unbiased screen for synergistic combinatorial therapies. And in order to do that, we did a very, conceptually straightforward but technically difficult screen in the biosafety level 3 lab with SARS-CoV-2 infected cell lines and identify relevant compounds. And we established the assay in the presence of low concentrations of remdesivir, and then ran a whole library on top of that of FDA-approved compounds that we already had ready to go. So the assay is shown here. It was quite simple assay based on cytopathic effect. If we infect cells, we use VIRO-E6 cells and later also KLU3 cells. If we infect them with SARS-CoV-2, we get cytopathic effect, so these cells shrivel up and die. If you have an effective drug, they don't do that. That difference can be read out with a really simple cell titer glow readout that measures the residual ATP in these cells. If the cells are barely there, there's less ATP than if you have happy and healthy cells. And in addition, we did a cytotoxicity control with uninfected cells that would allow us to see if the compound itself actually destroys the cells, which is also a possibility that we had to distinguish. Developing this assay, we were able to get really good Z prime of 0.6 in presence of remdesivir. So it's challenging to do these combinatorial screens because you lose window, but it was really well done. This was a lot of technical work that was really good that made that possible. So with this, we conducted the screen, and what we found is a busy slide. I'll walk you through it. Focus on this side. We have the entire library screened without remdesivir. So this is just your standard infected cells. What stops infected cells would be these compounds. So you see very, very few hits, and that's also consistent with what other people have seen. You see a couple compounds that are active. Nothing is quite as good as remdesivir. But the situation really changes if you have a small amount of remdesivir there. Suddenly you get a whole cluster here in red of compounds that on their own are actually inactive, but with remdesivir show strong activity and up to 100% inhibition of CPE. So that was very encouraging. That showed if you have a little bit of remdesivir, you add other compounds, a whole group emerges that is active. And this is the cytotoxicity control, just showing that these compounds do not kill cells in a non-specific manner. So they were dependent on the presence of remdesivir to see the effect. And then we confirmed these. So the previous slide was a primary screen. This is cherry-picked, tested again. They confirmed really, really nicely. So we had a high confirmation rate. And then an important experiment we did, and this was really spearheaded by Scott here in the audience, is to confirm in human KLU3 lung cells if the activity is there as well, and nicely confirmed. We focused, so this is the whole group of red ones from here, and the red ones on the right are really the ones that confirmed most strongly, and this is where we put our focus on, because we didn't want to go down a path of seeing effects in one cell line and not the other, and then ending up with something that doesn't really recapitulate in vivo. So in order to characterize these compounds more, we used a checkerboard analysis model that allows you to determine quantitatively if you have synergy or just additive effects, because that was important for us to distinguish. And we used this software, is available online. There's a variety of models I don't have time to go into. Suffice to say, we used the most stringent one for our analysis, and you would develop a checkerboard where you have remdesivir increasing concentration on one side, on the other axis, the drug you're interested in. And the software then determines a so-called zip score, and if it's smaller than 0, it's an antagonist. Between 0 and 10, an additive, and greater than 10 is a synergistic compound. In the follow-up from the screen we did, we were able to show that the approved HCV NS5A inhibitor velpatasvir from Gilead in our experiments was strongly synergistic with remdesivir. And we found a whole variety of compounds, but this is the strongest one and most pronounced one. So in this 3D plot, you can see velpatasvir on one axis, remdesivir on the other, and the dashed line is the single treatment, and you see not really quite modest effect. Velpatasvir on its own does barely anything. Remdesivir in this plot also, but the concentration is really low, so if you go higher, you see a strong effect, but we wanted to test at low concentrations. But once you add these low concentrations, ineffective concentrations of remdesivir and velpatasvir together, you actually get massively increased CPE inhibition up to 100% in this plot. And this is shown in this 2D display as well, where you have the dose response here of remdesivir, here of velpatasvir on its own, and then, this plot appears if you increase velpatasvir, showing synergy. And this is the derived data, so this is all the direct CPE data, but this is the calculated data in the Synergy's quantitation software. It shows a really nice peak, so you can see where most synergy occurs. And we were able to get a synergy score of 85, so it's massive. You know, most synergistic compounds, it's like maybe 12, 15 is what you get. This was really strong synergy. And if we slice these plots and just look at one shift of the dose response curve here of remdesivir, which was, you know, modestly potent in our particular assay, if we add valpatasvir, we get a shift that's about 30-fold shift in potency. This is still to date, to my knowledge, the strongest synergy with remdesivir that was observed. We have several compounds that were identified, but this is a really strong shift. So we got really curious about this, you know, hepatitis C antivirals, NS5A, nobody really knows what it does. There's no homolog in SARS-CoV-2. We wanted to investigate that further. And first of all, we want to see, is this a valpatasvir effect? Is it a class effect? And so we began to characterize the effect in other NS5A inhibitors. There's a couple of molecules. Elbasvir from Merck basically shows a similar effect. So far, all compounds that we tested for NS5A inhibitors showed this effect, so we're pretty confident that it is a class effect, but for valpatasvir is still the strongest one in our hands. So that was exciting to see, you know, that the synergy is present across a class of molecules. We also confirmed that it's present in the clinically used co-formulations because valpatasvir is not actually available as a single agent to patients. It is co-formulated under the brand name Ipclusa with sofosbuvir, and similarly the Merck drug is well co-formulated with another molecule and also works with the co-formulation. So here you can see the shift between remdesivir and then the co-formulation even a little bit stronger. So that was really exciting. And then, because all the data I showed you so far was from the screening assays, cytopathic effect, we wanted to do all the necessary controls to convince ourselves it's not just an artifact from the screen or something like that. And for that, you know, Scott pioneered TCID50 assay here showing, and it's a very busy slide, but I highlighted the important ones. Remdesivir alone at a low concentration that we've used, not much effect on TCID50s. Valpatasvir alone, also nothing happening, but together really strong, you know, several orders of magnitude decrease in infectious viral particles. Same is true if we look at the assay by immunofluorescence, staining N protein, and also by RT-PCR quantitating nucleic acids. And this panel here shows you the data for valpatasvir and the clinically used combinations, and here for elbasvir, same thing, the combination much more strong than the single agent in immunofluorescent, TCID50, and RT-PCR assays. So that was really good news for us, confirming that the effect is not just confined to our screening assay, but actually is also true if you measure infectious viral particles and RT-PCR and immunofluorescence. Next, we wanted to look at this in a bit more detail. Here's just some microscopy immunofluorescence to show again. We stained SARS-CoV-2 N protein in red. Blue is the DAPI. So you can see infected cells here in the DMSO. If you add valpatasvir on its own, it's indistinguishable like the DMSO control. Remdesivir low concentration looks less infected, but you still have a number of significant red dots. If you add it together with valpatasvir, it's very similar to uninfected cells, as shown here. And Scott then did a really important control for us, which was hard to do, but he stuck with it and did it. Technically difficult, primary human bronchial epithelial cells, because we had data before in cultured cells, but not in primary cells. So Scott was able to show that the same effect reproduces in these cells. So here is valpatasvir in the clinical co-formulation, remdesivir, and then together much stronger effect. And the same is true for the elbasvir example. So at this point, we were quite confident that what we see is real, doesn't really depend on the assay method, not on the cell line. We had data in 3 different cell lines, all checked out. These experiments always worked, had to be fine-tuned with the remdesivir concentration, but other than that, always worked. So we were left with this question, why do we see this, which target could you inhibit that makes remdesivir appear more potent? So the synergy we observed was really looking like a class effect for NS5A inhibitors. We were looking for homologs of NS5A in SARS-CoV-2, but nothing there, not in sequence, not in 3D. Couldn't really tell how it works. We knew though, and this is also where collaborators from Gilead gave us a lot of useful input. John Bilello is in the audience today. These are large molecules. They bind to other things. So we were thinking about maybe they inhibit a transporter like PGP. Velpatasvir does, we know that. Is there potential for less specific interactions? We were looking into that at length. I don't have time to talk about today. But the other idea we had was maybe it's a proofreader. Coronavirus has an exonuclease proofreader, and that, conceptually would make sense for this mechanism. And there was some data from early in the pandemic also about virtual screening, where people predicted with docking approaches that velpatasvir might bind the proofreader, but nothing ever worked in vitro in experiments. So it wasn't really pursued, but there was some, some at least hypothesis that it could bind. So we reached out to a collaborator at Columbia University who does exonuclease proofreader studies using mass spec as a readout, and we pursued this hypothesis. And indeed, we were able to show that velpatasvir in these assays does inhibit the SARS-CoV-2 exonuclease proofreader. So this is a mass spec of a substrate RNA. This is the intact peak, it's over there. And then here he treats with increasing concentrations of velpatasvir. And what you can see is that these smaller peaks are the degradation products. And you basically get less and less of that as you increase the velpatasvir concentration. And that was really exciting to us because it provided direct evidence in this reconstituted system looking at NSP 1410 complexes that velpatasvir in a dose-dependent manner actually inhibits this exonuclease proofreader activity in a mass spec label-free assay. So that made a lot of sense. We don't really say that this is the only mechanism that's possible. As I said, there's potentially others that could be considered, but the positive data in this experiment was really encouraging for us to see. So the model we propose to explain the synergy that we've discovered is that treatment with remdesivir leads to termination of RNA synthesis and also introduction of mutants, as was shown. And then the proofreader helps the virus survive this a little bit and change the situation back. And what the velpatasvir and NS5A inhibitors could do is to block this proofreader and thereby help kill the virus in presence of remdesivir. That could also explain why you don't really see this activity if remdesivir isn't there, because if you grow the virus under optimal conditions in a dish, it just doesn't really need this exonuclease proofreader. It can still happily survive even if you treat with high concentrations, and that's highly consistent with all the data early in the pandemic where people tried to use velpatasvir on its own against the virus and it was never really active. And there was a hypothesis it could be active, but it was disappointing to everyone that it's not active. And I think the key is that this proofreader here is not really essential for the virus. But it's a compelling model to explain what we've seen, a really strong pronounced synergy across the class of NS5A inhibitors with remdesivir. That's really encouraging. So the next step is to conduct in vivo studies, and this is where we hit the first real snag in this project, because NS5A inhibitors, pharmacokinetic properties are not that great, but they exquisitely potent molecules towards the hepatitis C target, picomolar molecules. So that's why it works so well, extremely well, against hepatitis C. But for us, if you think back about the synergy plots that I showed you, we would need maybe low micromolar concentrations of valpattosvir in the blood to really see effects in a patient or in animal models. And that's something that's difficult to achieve because valpattosvir exposure is low and the Cmax in the blood is in the nanomolar range. So we kind of stuck there. We can't really do a good animal experiment because we can't get enough into the animal blood to be able to see the proposed effects. But the next steps would be to optimize potency of these molecules towards the new target. Somewhat not, it's not a surprising thing in a repurposing campaign where you find activity against a completely new target, because originally these compounds were optimized for years against the hepatitis C target, and now you're trying something completely different. Of course, it's not working as well. It needs to be optimized. So optimization of potency towards SARS-CoV-2 target would be one step. The other step would be to try to improve exposure, and then also try to improve local concentration in the lung, potentially with an inhaled formulation would be an opportunity as well. So these are future directions for this project, but I just want to summarize. This synthetic lethality high throughput screening approach was able to discover a class of antivirals strongly synergistic with remdesivir. Just want to make clear, again, this is different from a normal combinatorial approach, because what everyone is doing is taking active compounds and then combining them. If you do that, you would miss these compounds. So this is really a synthetic lethality approach. You find compounds that on their own, like, have no activity. Straight up valpattosvir in SARS-CoV-2 has no activity, but with remdesivir it's really strong. So this is also showed a way to discover previously elusive exonuclease proofreader inhibitors. Many people were looking for those, but in the absence, at the time, in the absence of a biochemical assay, you couldn't really get them from a phenotypic screen where you just look at SARS-CoV-2 replication. In the meantime, a biochemical assay has been published, and also the compounds they found were also synergistic with remdesivir, which is interesting. A lot weaker than this one, but also, it was encouraging also to see that data. But I want to highlight this method also as an opportunity to identify completely new, otherwise obscure chemical matter that you can't easily find any other way. And so in this, as a last point, while the potency and pharmacokinetic properties right now don't support dosing, the preclinical development path should be a lot shorter. If you start with a highly extremely well-tolerated drug like valpattosvir, instead of starting with some starting point from, some fragment from a drug discovery campaign that was conducted de novo. So I think in principle this approach could save a lot of time should we choose to continue to optimize these molecules. And I just want to close with thanking everyone on the team. This was a fantastic collaboration with Sarah's lab, and then also Jingwei in Colombia. Scott did a lot of work, and he can tell you more details about the project, and many other people also on this slide. And we got funding from COVID Catalyst and from FAST grants. Thank you so much for your attention. I'm happy to take questions.
Hi. It was an exciting talk.
Thank you.
Have you looked at other variants of concern, or have you tested just the ancestral version of SARS-CoV-2?
Currently just the ancestral version one. Yeah.
Okay.
But I think Scott probably you have the new ones growing, right? We could look at that. Yeah.
That's exciting.
Yes.
Also, my second question is, have you observed any emergence of resistance to this combinatorial therapy over time? Like, increasing the concentration of the drugs over time and looking if there is any emergence of resistance?
I think we haven't really done that. We talked about it, right? But yeah, we haven't studied it long term. Mm-hmm. Would be a nice control to do that, also to see then if the resistance is conferred by maybe mutation in this in the target, right? So this is a study we were considering doing. Yeah.
Thank you so much.
Thank you. Good idea.
Nice
Yes.
talk. So I was struck in your initial assays, the curves that you were showing in Viros, Cali-3s, whatever the cell lines were, that your remdesivir curves were getting to 100% CPE inhibition, but the NS5A inhibitors, both alone or in combination, was not. And so is that also giving you a hint as to the mechanism, or do you have any sort of hypothesis
so
around
this
that?
is actually a feature because it's the NS5A inhibitors
Exactly.
on its own. It doesn't show any activity. Like even at 40 micromolar, it's not active because the virus can overcome it really easily if remdesivir isn't there, right? Only if you stress it with remdesivir, then you begin to see that it really suffers if an NS5A inhibitor is present, right? Because it can't do the proofreader function anymore.
It would be really interesting to look at entry as well as then just moving along. Obviously,
Right.
you're further along in the pathway when you're using these things. So that delta could be really impactful when you guys are looking at your different variants. So if you're
Right.
moving to other ones that have different kinetics in cell
Right.
culture, you might actually see more variability there. So something to keep an eye on.
Yeah, okay. For us, the CPE assay, I mean, it was, we incubated for quite a while, so I don't know if it was just, we didn't really resolve it kinetically really to that level, but this is interesting future thing we could do, yeah.
Thank you.
Thank you.
So thank you for the wonderful talk. Particularly, I like your molecular model. So I'm wondering whether you have done the experiment when you knock down the targeted proteins in the cells, and do you expect to see the co-knockdown of the different proteins will reproduce the synergy that you see achieved by the drugs?
Yeah, I'm not aware anyone has done it, but it would be good control to do also, you know, to take out the exonuclease proofreader, see if it still works, or if there's another mechanism, right, that could contribute, yeah.
In the back.
Yes.
Great talk. Is there any efforts for you guys to actually co-crystallize the drug with the target to, you know, to actually do SIR activities or, you know, studies?
Yeah, this is also a great idea. I would love to do that if anyone is up for it. Gilead, yeah.
And just another question. I mean,
Yeah.
Hep C drugs are not especially cheap. So have you actually found any other drug that has synergy that are not for Hep C?
Yeah, we did. So we have, in the paper we published, there's a whole list of things. Valpattizumab was by far the strongest, so it was really the front runner. But we found also some over-the-counter things. For those, we didn't investigate it quite as deeply. You know, it's difficult to think about. There's some encouraging data, like omeprazole was in there. There's some encouraging data in the literature that it works, you know, in infections a little bit. But I don't know. You have to really get deep into the biology, right, to also make sure that what you see is real. I personally got quite convinced after we had all these positive controls working, and then also the mass spec data. I like to have at least an idea of a potential mechanism. Otherwise, the combination therapies, it's challenging because, of course, a whole lot of other things could happen as well, right, to really convince you. But we published a whole list of compounds, also the whole primary deck, so there were several molecules that could be pursued if someone has bandwidth to do these studies to see, you know. But I think ultimately pricing, I mean, as synthesis improves, and I feel it's also a question of how many patients can you dose if you have a large market. I think these are ultimately more solvable problems. You know, if the data is strong enough that it provides a cure or a good therapy, then I think ultimately the finance problem is a bit secondary, although we talk a lot about it. But we can figure out how to make it cheaper, you know, I think, or more affordable. Yes?
Thank you.
Thank you for your talk. In looking for synergy, why not start with lead compounds that are known to bind to the other proteins in SARS-CoV-2?
Well, because at the time, this was super early in the pandemic, first of all, there was no comprehensive data like what would bind to other proteins in SARS-CoV-2, and then we really wanted to do an exploratory campaign, right? I think hepatitis C drugs, NS5A inhibitors, I mean, the data that they would bind to anything in SARS-CoV-2 wasn't really there. So it was very maybe a possibility, but nothing really strong, nothing done in biophysics, you know. But this is the beauty of screening, that you can actually find them like this, and then normally we confirm binding later, because it's technically also more challenging depending on the compound. Some do bind, but it will take you a couple of months to actually get that data properly. Any other questions? Then we're right on time for coffee break. Huh? Thank you.